so it's not a big deal to me if we don't find them. I mean, obviously, the finding of the live mussels here in the alpogenic carbonate of the seafloor is very significant, and I think that tells us something very important geologically.
Thought, since our goal
I'm
is
fine
to
with
find
whatever you feel
most
comfortable with on the Okeanos.
It would be crazy
it does sound like you're going to have to move the ship in any case since we're moving towards waypoint 2, which is to the north.
Okay, yeah, we can go back to the dense live mussel bed and do a couple more of the zooms. I'm sure, Bernie, you'd want that here, and we still have a little bit of time to do so.
Yes, if we have time, we can grab a few more of those nice live patches and get a few zooms on those. Thank you.
Okay.
And then just FYI, shoreside, looking at the sonar, which we have extended out on our current heading about 30 meters, the mussle beds continue, although they are much patchier, or at least the hard targets continue.
Sure.
But they are patchier, and they may continue beyond the range of the sonar at this point.
I won't name it yet in case
although
we go
it'll
further
become
north past it.
Much patchier.
That's good.
Well, that's great. Thank you very much for that information.
Watch lead, pilot.
Yeah, go ahead, pilot.
Hey, co-pilot recommended I drive over it in Cirrus view quickly to look for bubble reflections.
Sounds good.
Okay.
And then did you hear the other, we'll do a couple more of those partial and then macro zooms into the live mussels?
Yeah, let me know when to stop and do that.
Okay.
I'll do a quick drive across first.
So we're going to do a drive across now to the east.
You ready, co-pilot? You want me to do it right here?
Yeah, if you could push to the right on my screen and then come toward me, like out over the bed and then toward me. So to your left front. I mean, that will, anything that puts whatever it is between your lights and me is going to maximize its visibility, I think.
And the Chimera hydrolagus again in view. We're at a depth of 14, 18 meters. We've dropped several DVL targets too. And what's our temperature here?
4 degrees.
4 degrees C.
They could try a few zooms, quick zooms here to see if we look at a few of these live beds, please.
Okay, can we do snap zooms into the live mussels?
Sure. Sure. Any in particular, or just all of these?
To the right of the carbonate block, to the right of the rock.
Okay.
To the right would be fine. Yeah, we can start there and look around.
Screen.
Yep.
Tilt the camera down. I believe this is just a snap zoom while we're looking for bubbles.
Can you go in a little more?
In a little more video?
Perfect, thanks. How's that, Bernie, for right now?
Yeah, right at the base of that big rock, so it would be at 6 o'clock to where you're zoomed right now.
And if you zoom all the way out, the base of the big rock, they want to see that as well.
Okay, can we go out video?
So right on the left where the mussels meet the rock face.
Kind of where they're whiter?
Yeah.
Okay. Sorry, co-pilot, we'll do our bubble search in a second.
No worries.
And about 7 o'clock on the screen, on the #1 monitor.
Did you hear that about 7 o'clock?
Going to 6 o'clock now.
Oh, down at the bottom of my screen?
Right, right now. If you that patch, it's hard to say. Up the other way. It's kind of on the left side of the rock.
The dense circular patch?
Yeah, the dense circular
patch. By the
patch.
lasers?
Yep.
Okay.
That, Bernie?
Yes, that's fine.
Yep, that's it, Carl.
Okay. Come down a little.
Okay. We'll take a look at that then too. Yeah, you can partial in on that.
Okay, video, let's get a zoom.
Pushing in.
Keep going. Good.
Partial.
Thank you.
I'm gonna adjust my gains.
And then do you want to go in full?
Pushing in full.
Yes, please, Andrew. Thank you. See, there are a couple different sizes there.
We're seeing those caprellid amphipods again, the walking sticks.
Tracking focus.
Thank you.
Trying it.
That's the end of them. Can we go partial video?
Pulling out.
Yeah, we're trying to determine these little small white on the shells. These are amphipods as well.
Let's go wide.
White dots.
Oh, we're looking for white dots.
And can you then pan left? Roland saw something.
It's the larger patch. Upper left, you can see it's fuzzy.
Gotcha.
It's a quite extensive patch.
Up there?
Yeah, full
Kind
left.
of, there's a transition here in the mussels. Looks to be more dead ones where we are, and then to the left.
Zoom in on the transition as we go.
Yeah, sure.
Just science.
Yeah, it appears a strip of dead ones goes right up through the center, and then to the left there look to be much more in much better shape, and then to the right they seem to be in pretty good shape, but the left side seems it's
lots of very small.
Living a better life.
There looks to be a lot of very small mussels here. So can you, yeah, hold here for 30 seconds and then pan left.
Okay.
Do you want me to pull out for the lasers on this shot, science?
Wow, a lot of new young mussels.
Yeah.
Yes, please, sorry, thank you. Yep, perfect. Are there gastropod associates here as well? Okay, and then can you go full zoom?
Okay, full zoom video.
What is that white? I guess it was just part of the mussel. They're shimmering. Okay, you can come out.
Okay.
And then
Wide
just
video.
pan left and do another one, kind of where all those smaller mussels are.
Sure. How's right here?
A little more.
Okay.
Okay, we could come in, keep the lasers in view.
Okay, zoom to lasers, please, video.
Thanks, Carl. Thank you, Roland.
Okay, I'll try to steady up.
Good, Brian.
So we are zooming in on Bathymodiolus mussels,
Sure.
and there appear to be hundreds of very small mussels. We do have some larger sizes, different size classes, but the small ones are indicative of recent recruitment event. And then we can zoom all the way.
Okay, full zoom.
A small shrimp right at the top center.
Nice focus video.
I'm sorry, Bernie, you saw a shrimp?
I think it's just a small shrimp that was in the top center of that last, just a few seconds ago.
Okay.
Just above the dark spot, right there at 12 o'clock. Maybe just an amphipod. Sorry.
Another coprolid. Yeah. Okay,
good camera work, ship size.
Yeah, excellent camera work. Are you happy with this zoom, Bernie?
Yes, I'm fine, and
ahead,
we've got what we need with this, so they can zoom out as needed.
Okay, so what we're going to do is zoom out, and then we are going to move over the mussel bed, do a search over the bed, look for the bubbles, and start to get set up for our 2 o'clock live interaction. But we're going to stay here and work this area. I think there's a lot that we can zoom in on for the public, and we'll continue to work this for a little bit. Is that good with everyone back on shore?
Go ahead, Survey.
That sounds fine with us. Thank you, Andrea.
So, Andrea, it's Carolyn. Is the intent to use this site then for your ship-to-shore outreach effort, or are you going to go up to waypoint 2 for that?
We're going to use this one for at least the first outreach effort, but we'll definitely be moving up as the day goes on.
Okay, thanks.
From your view of the points to the north, is this mussel bed more massive than what we saw north?
Bernie, you've looked at those as well. I would say, I think it is. Like I said, we hit it near the end of the dive last time. This one is actually quite similar in character, but I believe this is more extensive.
I'll drive over to the eastern extent.
Bernie, do you have a...
Could you give me...
Yeah, this is much more extensive. We know...
Copy that. Ought to drop
this one has a lot more of the younger, the smaller size class mussels. This is a very good one. I think this will be fine, and then we can go to the others as time permits for the other interactions. But this will be a great one.
Is you can come to the ship.
Yeah, and just a reminder to the ship. I know you've seen this before as well because you've been associated with these dives too. But one place that we often try to look for methane, if we find any indication, is sort of in these underhangs.
So that's a game we can play, and then we can
I haven't seen anything, and I would assume you would see it on Sirius. I did want to ask the ship though, if you have hard targets like carbonates, and then sort of targets in the water column, is that really that easily distinguished on the sonar?
Left, if that's okay.
Yeah.
Did you hear that?
Let me just double check.
Can we distinguish water column anomalies in the sonar?
Watch
yes.
the pilot.
Can you distinguish between the carbonates and the water column anomalies when you're looking at the sonar?
Probably one of your better people to answer that question might be Tom.
We're having a conversation back here, Roland. Thank you.
Ship move late in, Nev?
Sorry, Tom, what was that?
We can, not just directly with the sonar based on what the sonar is saying, but what we can do is bring the vehicle up a bit. And that's what we were just talking about with the pilots is so Tom was suggesting we fly a little bit higher to where we get the ground targets
so
out of that fan.
There.
You know, it's a, I don't know, 20, 30 degree fan beam. So if we come up high enough, we'll get the ground targets out of that, and then we'll be able to just have water columns. So that was what the co-pilot was discussing.
Auto XY.
Right.
Yeah,
Thanks,
that might
Brian.
be really worthwhile for a few minutes if you've got the time. Thank you.
And pilot, co-pilot, Carolyn Ruppel back on shore was just saying that at times the bubbles are coming out of, you know, bare patches in the middle of the mussel beds.
Copy that. Thank you. You're still floating left, Carl.
Yeah. User error. I didn't hold the auto XY long enough. Then I hit the other two. I was just going to hit it, so if we float up for a couple minutes.
Yeah, it doesn't have to be real long, just long enough to get a couple of good sweeps on a constant heading.
Yeah.
If we get any
You
bright
just have to wonder if the organisms are generally intersecting the methane before it gets out for the most part in a place like this where you've got so much biota. It's pretty amazing.
Here.
Okay, there we go. Okay, I've got my heading and XY locked in.
Okay, we are facing 30 degrees is our heading.
Coming up.
Coming up.
We're coming up a little bit.
Great. Yeah, I'm going to bring it down to 20.
Delta is still 12.
There's something. You go back down, Carl.
Go ahead, Bridge. Thank you.
Back down?
Yes, please.
Okay, pilots,
Didn't
the
see
ship is
anything?
holding position.
Nothing definitive, but there was something right here. If you go back down, I'll see if we reacquire it. Copy that. Thank you, Nev. And then, yeah, like Brian said, we'll just push you out that direction, turn around, come back.
Okay. Yeah, it might have just been a fish or something, huh?
There was a fish center screen ROV now that was a little higher in the column.
We're just taking another survey of this mussel bed and keeping our eye out for methane bubbles coming out of the seafloor.
Nothing definitive.
Okay.
Okay. So if you can drive out in what you feel is the middle of it, Carl.
Yeah.
So you hit the end of the tether. And then do a 180 and come back to me right down the middle of it. That'll backlight anything that's there.
You bringing your heading to the strike about?
I'm going to try, yeah. I'm coming down a little too.
Yeah, I think it's about 040 or so. 035, I don't know.
Local time is 13:15. We are diving New England Seep Site 1.
Not a lot of life here. There's still the carbonate, but
The Chimera. Huh.
okay. What's your camera angle?
Looks like there's corals here.
Yeah.
On the right.
Yep, some nice corals there.
Yeah, so you're coming to the end of your tether there.
Okay. Nav, do you want to drop a target, remove your northern reach target to here?
Sure.
Maybe note there's 3 nice looking corals. Okay.
I wonder if this is the same
those
site
might
actually
be, yeah.
Where we documented the corals before.
Yeah, that's that target right there, probably.
So.
Or something associated with it.
We're back to that, huh?
Yeah.
I believe that wasn't that further to the north?
Yeah,
that was north of 23 on the waypoint map.
Based upon our waypoints, this looks like it just is an extension of what you all had discovered on the last dive. This furthest most extent that we're just dropping a waypoint is probably 12 meters or 15 meters from the red and white coral and discolored sediment. So,
all right, so
excellent.
Down
There's at least, I counted at least 8 colonies as we were going over that. Nav, could you add coral to that waypoint too?
Okay.
Sure.
Thank you.
Slide to port a little and just do a real close skim over all of that community there.
Okay, I'm about centered.
Yeah, looks good.
Come down a little more.
And again, the image in view here, a dense mussel bed off to the right and carbonate outcrops on the lower left of the video.
All right, come on in. And I'll pick up as you get close.
And we are headed back due south. We're at the northern extent of this mussel bed.
Yep, sounds great. 1.5 meters up.
And we're 1.5 meters off bottom. Our depth is 1420.
It's weird my lights don't show up symmetrically.
One of them might be farther up than the other.
Yeah. Okay, we're driving south down the strike of the mussel bed. Trying to get bubble reflections.
It looks like... Oh, from Sirius. 40 meters, yeah.
And we're running south. What's that noise?
This is the highest point of the...
Okay, we're reaching a high point here. That sounds good to me. Yeah.
I'm not opposed. You want to go back where we started, Brian?
We're moving south, 1420 meters.
No, we did.
We
go
did.
all the
We
way
got
to the
southwest
extent.
We didn't go over and tag the waypoint itself. Sure. You want to just move Sirius right to it? All right. Let's do that then.
Okay, copy that.
So we're done our bubble search?
Yeah, just keep moving south along the strike here, Carl.
So I'll bring
We might get lucky.
Yeah. Okay.
So if everyone agrees, I'm going to bring us down 40 meters.
Another coral.
205.
Let's aim for waypoint 1 instead of southwest edge of muscle bed.
Sure. So that
I think that's what Brian had in mind. Okay. All right, so what you said then, Nav.
Okay, I'll put that in.
Antamora.
Bridge, this is Nav. Hi, we'd like to request a ship move. 40 meters at 205 degrees and 0.1 knots. Yep, that's correct. Thank you. Okay, ship move is underway.
205.
205.
Copy that.
Continuing to run down south through this muscle bed.
Theo, can we get a snap zoom on this?
Yeah, an octopus.
Okay, that's good. Full wet. Just double checking.
Can we zoom back in there a second on the white material that was to the left
we
of that?
zoom back
Never mind,
into
you're
the
going too fast. Never mind,
octopus?
don't worry. Sorry.
We can do it, Carol. We're right here still.
Interesting
swirl.
Living thing.
Creatures.
See that white material under that ledge there? Swing to the left.
Did I hear back in on the octopus?
I'm sure it's just.
Can you zoom back by the octopus?
Just like to
Okay.
zoom in next to that octopus there. You see what I mean?
Uh-huh.
The white.
What's our ship move now? 0.2, right?
Sorry, I didn't get you fast enough there.
No, we're
Less
fine.
than
We haven't moved too
two
far.
tether links, so
Okay.
you should be able to just do whatever you want to. Well, not whatever, but you know what I mean.
Taking off.
Won't go far.
Yeah, we want to look under the ledge to the left of the octopus.
Okay.
Because we want to scare him.
Video, let's go in.
Yeah, that's just Matt. Okay,
Okay,
thanks.
You can come back out. Thanks.
That was all, watch lead?
Yeah, we were just looking for hydrate.
Okay.
That mound straight ahead. Should we look at that?
Are there kind of searching?
You want to do a snap zoom?
No, that's okay.
We were just looking at this mound.
Huh.
Just thought
It's like a mound of mussels.
It was so different, we thought you guys might want to take a snap. Just to see if there was interest.
Numerous small ones, several size classes here. What's that? Oh. Oh, never mind. Yeah, can you zoom in on this ledge in the center?
Sure.
Black ledge, your command?
No, let me get around, pirouette around.
Probably want to see the fuzzy just below it.
We're at a depth of
Okay,
1422. Local time
here.
is 1327.
I think they're just looking for the hydrate ice.
That would collect there. Oh, that clam just closed.
That's good, thank you.
So we're very close to the southwest edge. What was the plan after we got here?
I think they wanted to make sure they touched that mark. Oh.
To the west. Okay. I'll just make sure we're fully off the southwest edge. What was that? Oh, sure. There are some more rocks ahead of me. But gotcha. Oh, the actual multi-beam target?
Chimera, likely the same individual we've been seeing since we've been at this muscle bed. Do you want to circle back around waypoint 1?
Yeah, let's follow this ship move. Still got a little bit of serious to go farther south. All right, Carl, if you want to come back on those rocks.
Oh, you're done with that? Okay.
Oh, we're supposed to switch watches.
Yeah.
That'll be fine.
Back to the rocks.
Would you get him? Did you find them? Oh. It's a black dogfish.
Hey.
And we're back at the target we dropped, the southwest edge of the mussel bed. We can circle around.
Hey, Dave. Hello.
Hi, Brian. I think it's right in front of me, or at least warrants a closer look. Okay, well, anyway.
So Dave, what we've seen so far is, we're at the southern end of this mussel bed trying to find bubbles. I've not found any bubbles. There's some good live mussels around this sonar target on HiPAC. There's a little mound, just, the more vigorous biological growth. And then there's a particularly nice patch up here to the north where it says live mussels and juveniles.
Thanks, Carl.
But we've been down through this axis a couple times looking for active methane. And
have you gotten right down on these, looked under these rocks?
We just got here.
Just got here. Before I head back north, instinct tells me I should squint down here and have a look around.
Right, we came into a little bit to the north, and we turned and looked at it just when we came down there, and there's certainly no biology there. Yeah, my sense is we did not fly directly over it, but we watched it go by.
Yeah, we came in and saw the mussel bed and kept going.
Right.
We came in a bit to the north of waypoint 1.
My memory is correct. Last time we were here, there was a solid patch stream of bubbles.
We haven't been here to this site before.
Really?
No.
It's
not
south.
to this end of it.
Yeah.
It's
this
south
is
of
south.
where we were.
Okay, that makes sense. That makes sense. Okay.
No, there's a solid stream of bubbles on the north end of this dive, about 800 meters away.
Okay. Yeah. Because I was looking at the old photos and GPS locations last night, and the bubbles were way up north.
Up there. Yeah.
Yep.
There's a crab in view. Can we get a snap zoom on that before we continue on our bubble hunt?
On crab upper left.
Go ahead, Bridge.
Not interested in crabs, Rolly.
Yes, she just called it.
Oh, I didn't hear that.
Science, did
There's
you
a
just
fish in there too.
call up on pilot, ROV pilot? He did not hear your call.
Yeah, I don't
You're not listening to science, I don't think.
Science, did you press the pilot button when you requested that?
Did you turn me off, Dave?
Did you press pilot when you did that call?
Oh, no.
You have to poke the pilot to get his attention.
I know, but I guess I just was in the mode because Carl was just listening to me. I wasn't holding down, so.
There's a crab, top center.
2 crabs.
Okay, Rolly, we can zoom us a bit. That's good right there, thank you.
2 crabs. They do look fuzzy.
Okay.
Cyclothony.
So I'm just about right on top of you, Dave.
Yeah, that's fine. I'm gonna take a real good look around here before I move north.
That's fine, just letting you know.
Because we've been right in the middle of this stuff before and not seen the bubbles until they were
you got to be
literally right in front of our nose.
Yeah, you got to be right up on top of them when they're these small plumes, I think.
How are you today, Dave?
I'm asleep at the wheel, Andy.
Lack of sleep last night from a lot of us.
You know, it's a little bit beyond rockabye, baby.
Yeah.
So when I see these bacteria on the underside of the rocks, it's a sure sign that if bubbles aren't here now, they were at some point. It's been true everywhere else we have seen bubbles. So I'm just hoping one lazy bubble will rise for us from here.
Is that a fish next to your lasers?
Yes, it is.
Oh, Gattyrapsaurus under
here,
the ledge.
Oh, that's good. Focus fish. You see lots of bacteria on the between the rocks here.
Bridge nav.
Large patch of dead mussels.
Copy that.
Here,
Thank you, Bridge.
and a lot of bacteria, white filamentous bacteria. And the pinkish fish is a Gattyrapsaurus, or a type of rockling. We are currently at a depth of 1422 meters, and our local time is 13:39.
Another crab in the
I see that.
crevice there. Okay, Roland, let's come wide. Let's back up a smidge here. Okay. All right, I'm gonna work my way a little bit more northerly. Facing north presently, back along the axis of the activity. Staying to my left, to the west.
there were bubbles in the multi-beam. So we were hopeful that there were bubbles.
All right.
Yeah, look at all of the little,
just
small young mussels
around,
on the edge.
No, we have. I mean, we have not gone to the very far southern, but given we got 20 minutes left, I think it's probably good just to find something nice and
so you're behind me now.
Set up and.
Yeah.
I'm at 90.
I'll be turning around, be able to turn around and watch me as I go here. Roland, let's get a little zoom here and let me have a look at some of these
Bacteria streamers.
Yeah.
Co-pilot, I believe we can turn around and face the other direction.
Okay. I'll just have my back to our direction of travel.
We're going to head north.
Oh, we are? Oh, okay. Sorry, I misunderstood.
That's a partial zoom.
Yeah.
Setup is
bacterial mats here, abundant here. Yeah, chipping. And our depth is 1421. We're getting a video of white bacterial mats and seepy sediment on the seafloor.
Andrew, if it's not too much trouble and doesn't interfere with what you're doing, if you could focus on one of those mats that has those super white patches, because that's been successful in the past. Like the one over to the right on my screen.
Sirius just a hair north.
Can you zoom on Dave?
Yeah,
give you lots of active
on
targets,
right,
Dave.
on the bottom.
Yeah.
That's fine. You can get that going. Just gonna.
Yeah, the one right in the middle.
Something going for science.
Yeah,
here.
Roger, the one right in the middle.
Coming down.
Yeah, that would be fine.
Okay, a little bit of
zoom,
please, roll.
The one over to the right. Either one is fine.
Bridge RV nav.
Partial.
Yeah, this structure looks a bit different than the filamentous bacteria.
Yeah.
Well, if that isn't a likely suspect right there, but...
Bridge RV nav.
I think you have to get me closer. That's max zoom.
Yeah, that's...
I can't tell if it's sponge material or what.
No, it's a...
Very pillowy.
Amanda's word is marshmallowy if it's the same stuff.
That's right, marshmallowy.
Bridge ROV nav.
Okay, come wide.
All right, I think we're going to have
Bridge,
to get
we'd
set
like
up
to request
here.
another ship move.
Yep.
15 meters, 025 degrees, 0.2 of a knot.
Okay.
Copy that.
Hey, Andrea, this is Tim.
Hi, Tim. How's it going?
Hi, good morning. It's been great. This is a great site.
Okay, I'm going to move myself laterally to the
Is it okay if we talk while you guys are setting up?
Absolutely. I'm going to have to go into the forward lounge in a couple minutes.
Let me just interrupt for a moment. We're going to take the engineering stream off, and it's going to be converted over to the live feed.
Okay.
Well, I guess I was just wondering if Bernie is still on the line.
Yeah, I'm back up here. And I just got back and getting a bite to eat. So I'm sorry. Thank you.
No, that's okay. I wanted to ask your impressions of this site because I know you've seen other sites in the Atlantic. You've seen the Blake Ridge and whatnot probably too. And so I guess I was just wondering your impressions about this site, what you're seeing that's similar or different. Just get your impressions.
Well, just from some of the sites that I've seen, I've only been with Cindy for about 18, 20 months now, but the sites and from that I've seen, this is really very good because we don't tend to see get all of these different size classes as abundantly, especially as we have at this particular site. It was nice to see some of the scale worms and along with the amphipods and a few of the other associates.
Can we drop a target, please? I'm sorry to interrupt. Can we drop a target where we just saw that white material, please?
Sure thing.
Or close, wherever you are now is close enough. Thank you. Sorry, Bernie. Go ahead.
No, that's perfectly fine. Please stop us if we're going on. You have to get something. I haven't seen as many shrimp in here as I think we've seen in along some of the other beds. But this is a beautiful site, and I'd really like to come back here next year and pick through this.
Yeah, no doubt there'll be some great samples, for sure. There's a lot of invertebrate fauna in here, and I agree. I haven't seen any shrimp, and I think the Seep site we went to earlier on this cruise, I think we saw one Seep, literally be a Seep endemic, a shrimp that only lives at Seeps, on that dive we had. But anyway, I was really curious of your thoughts.
Yeah, but I think Cindy was logged on earlier from her holiday location, but she's surely going to want to look at this video. This is really beautiful.
And you guys are coming back out in the spring of 2014, right?
That's the plan so far. Yes, it is, and hopefully Blake Ridge as well. I think shortly thereafter, I believe.
Yeah, Tim, I talked to Cindy last week. This is Carolyn, and I'm sure Bernie knows all of this. And so, she had a cruise already funded by NSF to go down to Blake Ridge and revisit some of those features that she's been to several times. But NSF did add days to allow her to visit the Virginia and Nantucket Seeps, although who knows whether it'll be the same cruise or a different cruise.
Well, I certainly hope you guys can get up this far north and be here at this. This is a great site. Lots of really active areas, not so active areas, some dead areas, and then there's certainly it's patchy for sure with these white filamentous microbial communities on the mussels, but also patchy in terms of recruitment. There's one area that I noted that was remarkably dense with new mussel recruits or recent mussel recruits. So really a fantastic site, and it's not so huge that you can't map it and be manageable. It's also sort of good news in terms of trying to characterize and quantify what you see.
This would be a nice site to have Century do some nice imagery for us, put together some photo mosaics, and then actually be able to piece through this, look at the sites, the areas that are patchy, and try to see if there's anything we could start pulling out any information as to why the distribution is like it is, and especially in relationship to the matting and some of the other areas with carbonates.
Yes, those of you he mentioned Sentry. That's an autonomous underwater vehicle, basically a robot you program at the surface, or you can program it, I guess, now in midstream acoustically. But you can have it run track lines and do very detailed imagery of the seafloor, having it hover through a navigated network, and it's a fantastic vehicle. Bernie, are you guys going to have Sentry when you come back out?
I know Cindy's put in for it. I don't know if it's going to be available, but I think she's requested Sentry to be out there for these.
Yeah, that's great. I also think that some ROVs like Jason come to mind with being able to do holding position and hovering very well, and being able to do downward imaging and mosaics. I've seen some really good mosaics from Sentry. Again, it can well navigate itself in autopilot and run tight lines, and there's a nice squid going by. But anyway, yeah, I think an ROV could probably accomplish those same goals if you needed it to.
Well, yeah, certainly, because we'll be doing plenty of sampling. I think if we just have the ROV only, like as Jason, and we would just want to be doing, there's a nice squid. We'd want to be doing as much of the sampling and get some good footage here for the mosaicing. I think the big selling point for Cindy to get the Sentry out is often it can go out and do all the exploration or any of the real fine detail work before we go out, and that gives us we can pretty much pinpoint where we want to sample. But if it's not there, yeah, it certainly would use whatever we have at hand to its fullest capabilities.
That sounds great. So you guys will be sampling for community ecology, I'm sure composition and diversity, right? And you'll also be doing some genetics on the animals too?
Yes, that's we've been really doing a lot of the genetics work as of late. Sophie's been doing a lot of the work with the shrimp that we've collected from some of the Caribbean seeps, Mid Cayman, and a few from Barbados and some others in Mid-Atlantic Ridge. But Sophie's been doing the work with those, and we're trying to do the genetic, the population work through the genetics as well now.
Great. Well, it looks like you have some mussel populations here you could work with too. Plenty of them.
Yes, and this really has again with the different size classes here, and it's so wide ranging that this would be very. This is sort of what we've been looking for everywhere we go, if we can see this, and this is one of the best ones we've seen that I've seen.
So will you be doing population genetics on the size classes?
Yes, if we can sample these like that, yes, we would certainly do that. That would be that'd be something I think Cindy would be clapping her hands over. She'd be very, very pleased with that.
Yeah, it sounds great. So I know we got about, 5 or 7 minutes before they start this thing, so I don't know what. I'm assuming we're going to be on a different channel than everybody else, so we can keep talking, but I don't know.
Yeah, I was understanding that we would be a separate channel as well, so until we hear someone cut in and tell us to pipe it down.
Right. Andrea says correct. Hate to take that chance, though, and step on top of something, that's all.
Hey, Tim, it's Brian. You guys are clear to keep doing science on OKEX.
Right. Thanks, Brian. Yeah, at some point I'd love to sort of run along the carbonate here and see what's on it, too. We see some nice corals at the northern end, so it'd be pretty nice to take a look there, too.
Yeah, OKEX party line. Do you guys copy? This is Andrea.
Hey, Andrea, it's Tim. Copy.
Yeah, I got you, Andrew.
And if you can speak a bit louder as needed prior, that would be helpful. And is it Anne-Marie? Anne?
Dave, you're good with that position? FYI, I hear him coming through their speakers back in the microphone.
Yeah, Scott, this is Tim. I don't know if you were on the line earlier when we saw a few corals on these carbonates, these outcrops that run along the eastern margin, more or less, of the area. But there were, I saw Paragorgia for sure, maybe some other species. I know some point we'll get there. It's just to the northern end. It can't be more than, I don't know, 20 meters away from here. Maybe 30. Yeah, I just want to confirm too, have you guys seen clams? Because I don't think we've seen clams yet. Maybe we just missed it.
Just saw a few disarticulated shells that are distinctly clam, and they were of a good size, but I'm not, I really thought that we'd see some clams in this area, but I haven't. Clams we were seeing in the seeps down around Barbados, they were very, very highly visible. They were completely up out of the sediment practically, but I don't know if the clams around here are going to be buried or if they're just out further out from this location.
Will we hear the questions on the other end? Will we speaking directly with them? Great. And it's Annie who's hosting? Is it Annie or Jamie hosting?
Are we doing that all on series? Or is that below? Okay.
We're about 30 seconds away from the intro. Quite clear through their microphone.
Hi.
Hi.
Good morning. Absolutely. So NOAA, or the National Oceanic and Atmospheric Administration, is the federal science agency that's responsible for the understanding of the atmosphere and the oceans. And our mission really includes understanding and being able to predict changes in the atmosphere, the climate, the weather, the oceans, and the coasts. And to be able to share that knowledge and information with others. So that's exactly what we're here doing as part of this Northeast U.S. Canyons expedition. We're exploring unknown and poorly known areas and submarine canyons off the Northeast U.S. coast. So that's from about south of Rhode Island all the way up to the U.S. EEZ. My name is Kelly Elliott, and I'm the expedition manager for the 2013 Northeast U.S. Canyons expedition. And my job is really to work with the mission team and the broad ocean science community to develop the plan for the expedition and to oversee the planning and execution. And I'm here today on board the ship with our onboard scientist, Brendan Rourke.
Hi, I'm Brendan Rourke, a assistant professor at Texas A&M University. I'm in the Department of Geography and also part of the Ocean Drilling and Sustainable Earth Sciences program. As Kelly just said, one of my roles here on the Okeanos is as onboard co-leading scientist. So my main function is to integrate what goes on here on board the ship with the ROV crew and operators with the large group of scientists back on shore. Of course, one of the things that we're doing here is exploring these canyons along the U.S. East Coast, and what we're trying to do is quantify the biological communities, looking particularly for deep sea corals and sponge communities, as well as looking at deep water seep communities, as well as characterizing the geomorphology of these canyons. What we're doing is gaining baseline knowledge. We can further study these canyons down the road.
David, will you be able to get a zoom on the white coral?
That's a great question. So as the ship is transiting across the surface of the ocean, we have a couple of different sonar systems on board, and we actually use sound to image the seafloor. So as the ship is going across the surface, we're actually sending pings of sound down to the seafloor. And we measure the amount of time that it takes for that sound to bounce off the bottom of the seafloor and come back to the ship, and also the strength of that return sound. And with that information, we're able to determine how deep the seafloor is and what the seafloor is composed of, so whether it's more of a rocky hard bottom or something that's a bit softer and more muddy. So we take that data and information to create maps. The special and unique thing about the multi-beam sonar on board NOAA ship Okeanos Explorer is that we're also able to look at what's happening in the water column. So that's the area between the surface of the ocean and the seafloor. With our multi-beam sonar, we can actually detect schools of fish. We're able to see gas seeps like the one that we're investigating here today, and we're actually able to detect vertical migrations of some species. So we take this sonar information or the sound information and create a picture or a map of the seafloor that scientists use to identify areas that they want to investigate further.
So one of the things that we're particularly looking at today is we identified bubbles in one of the on one of those mapping cruises, and this has been in conjunction with USGS scientists in Woods Hole, and those bubbles typically indicate methane escaping from the sea floor. And so what we're doing today is ground truthing where we've seen those bubbles, and what we've found are chemosynthetic communities. So what these communities are doing is utilizing that methane to produce energy, and you have sort of a succession of different types of communities, starting with clams or mussel shells, and then growing outwards. So the mapping served as a target to then utilize the ROV on board the Okeanos Explorer to then go out and ground truth, and we have identified these communities here today just for the first time.
Thanks, Brendan. And that's a great example of sort of how systematic exploration works. NOAA's ship Okeanos Explorer is the first and only federal vessel with a federal mandate to systematically explore the world's ocean. And the first step in doing that is to map an area of the seafloor. So for the past 2 years, Okeanos Explorer has been transiting up and down the continental margin, creating a map of that area and looking at submarine canyons and identifying anomalies like these gas seeps that we're now investigating further during this expedition.
And of course, critical to our ongoing endeavors these days, or on this expedition, is the use of the remotely operated vehicle Deep Discoverer, or D2 as we like to call her. This is a brand new ROV. This is the first expedition in which they've been conducting scientific dives. It's a deep water capable ROV, capable of going down to 6,000 meters. It has 2 manipulator arms, over 6 different cameras, and a slew of lights to help image the sea floor. And I'll tell you from my experience with other ROVs and submersibles, the lighting system and the cameras and the combination with the videographers on board the ship, it is one of the best, if not the best, imaging system I've seen in use for ocean exploration.
Shoot us up a little bit because of the heat. Let's full wide on serials. Okay, copy that.
Yeah, absolutely. One of our other primary capabilities is telepresence, and that's how we're actually able to speak to you here today. So NOAA's ship Okeanos Explorer is a telepresence outfitted vessel. It's purpose-built to conduct telepresence operations. So with telepresence, we're able to stream live data and video feeds off the ship to satellites and then down to shore, where scientists, students, and the public can actually watch those feeds in real time. And what we're also able to do is have scientists join our ongoing exploration from shore. So they actually tune into those live video feeds. They're able to download the latest data sets that we provided to shore, and they can pick up the phone or use an intercom unit to speak directly with the scientists on the ship, log into instant messaging, and communicate back and forth via instant messaging about what the observations are that we're seeing on the seafloor. So through that, we can expand the number of scientists that we're able to have on board the ship from 2 to basically an infinite number. And that's really important because we're an exploration ship. Our mission is to explore the ocean. And since we're exploring, we don't know what it is that we might find, and therefore we really don't know what it is that we're going to find, and we're able to bring experts from all over the world and to provide assistance, whatever it is that we find.
And that's an excellent point, Kelly. We've had anywhere from 10 to 40 scientists participating, and it's one of the things that I love about this program. The ability to have an expert on call and the amount of questions that one can generate during an expedition like this is phenomenal.
Pretty good though. I can zoom in and show it then if you can stabilize. Tracking view. Just check over something. Keep going. Slow. Full line.
Thank you.
Are we done?
Do you want me to reestablish the audio and transmission?
Roland, are they done up there?
They are finished, but we're standing by for a moment.
You come wide.
I was talking to you for lunch. They did finish with our live, but just standing by. Okay.
Bubbles. Bubbles? Bubbles. Looking for bubbles. Nothing on the bubble sled.
We're just checking to verify. You heard him say that their live is finished. Yep, agreed. Right here, Rolly. What is that? Stuff. Oh, it's just setting shelves. Okay, wide again, please. I've been straining my eyes fairly, fairly hard, and I really haven't seen any visible those.
Possible that it's one at a time.
I haven't seen any visible. Yeah, pretty.
Okay, can you hear me, Scott?
Well, just on the far side of the rocks.
And can I confirm that you have the schedule of them all being 10-minute interactions? Our schedule said 20 for the first one at 2 p.m. and the last one at 6 p.m.
They're not terribly vigorous.
Can you hear me, Scott?
Okay, directly below your laser is also red. We just set up normal wet.
No, they've been looking for him.
Northward.
They're doing the Grand
Copy that.
Pacific interaction.
North, eh? Well, I'm gonna work my way
So
out.
they're doing live interactions pretty much every hour on the hour.
The axis of this mound is 0 to 0 is your.
We hear you. Our scientists are getting back in position.
And then Andrea and Brendan are rotating in and out so that one of them can still be here and maintaining the.
So you want to reestablish that audio jack?
Who's like the people that are interacting?
RIOE, Okeanos did hear you.
Whoever comes to the aquarium today, so that's really cool. As part
Big
of
red
hosting
coral.
the National Forum for Ocean Exploration, the Aquarium in the Pacific set up an exhibit that will be there for the next, I think, like 4 years, that is basically about ocean exploration. So it will have live feeds all year round from the 5 ships, the 12 designated ships. There'll be
Well, there's a.
We have a scratch for an audio check.
Nah, that's.
a bunch of specimen collected at depth.
I wasn't going to say that, but that's exactly what I was thinking of saying.
Should we talk about OKEX?
It's a filamentous deposit upon the drawer.
The idea is to sort of get
Gotcha. Thanks, sir.
involved and invested in ocean exploration. And it was their 20th this
forward launch. This is video. I believe I hear you guys transmitting.
So.
You close the mic.
Yeah. Just get her going before I use it all up. So these corals we've found are all on the east faces up current of the clam beds.
Makes sense. Can give you a little more leash, not much.
Thanks for thinking about me, Josh.
I'm always looking out for you, Dave.
Well, that's a big old thing.
Bubblegum?
I don't know. There's a lot of pink ones. Okay.
Yep, you're pulling me now.
Yeah, I can feel it. So need to get on the other side of this so the current's taking me into it. It's just prettier on this side. It looks like a Japanese woodcut.
Yeah.
This is Okeanos Video 2 on the Science Channel. Just seeing if Shoreside could talk to us on the OKEX party line or on the conference call.
This is Duke Marine Lab. We've got you loud and clear.
Thank you, Duke. Thanks, Roland.
Hi, Roland. This is Taylor Heil at Hui. You read you loud and clear.
Thank you. You guys are all coming in well here.
We're getting a lot of choppiness with the video feed on the number one. And the chat room, looks like several other locations are getting the same thing.
Okay, I'll check into that. Thank you.
Yeah, we're having the same thing on I2 here at Hui.
You say I2, you're talking the second stream or Internet 2? The home bandwidth.
Internet 2 on the first feed.
Copy that.
There's a little bump of carbonate. Doesn't have much bacteria on it, but
yeah,
why
look
is
at
it
this guy
sticking up there all by itself? Oh, I see that guy too.
And he's gone.
Called out for something. Transmission is getting spotty back on shore.
Okay, that's all right. I'm just surveying around. I haven't gotten terribly excited just yet.
Hello, everybody. I'm back. This is Andrea Quattrini.
Okay.
How's everybody doing?
Simply marvelous.
Have we imaged the corals?
Maybe not so well. Jason,
this little.
I'm getting
right
feedback. I don't know why.
I'm trying to.
Okay, maybe not so well. Jason indicated that he's gotten booted from the chat and can't get back in. I don't know if others are in the same boat or not.
There's a lot.
Yeah, I'm not in the chat right now. Sometimes we lose our chat room. That happened to us yesterday, so it might take a little bit to come back online. So right now, for those of you, we are not in the chat room.
Coming at me, Josh.
So you are welcome to call in. We can still talk on our party line. This happened frequently yesterday.
Oh. I'm back on the chat. At least I'd been off for a little bit.
That's a lot of clams. Bivalves.
I have a question. I wasn't here. I was coming back up from the forward lounge. Were we able to image the corals successfully and get close-ups of the brittle stars on them and the other associates?
We did not look at the corals.
Okay.
We imaged one, I believe it was a bubble vein. Small.
I can still get to it. I'll take a sweep around it before I leave here.
All right, I am back on the chat. Tim, are you still on the line?
I'm coming down. Yeah.
There's a little fish in there too.
Yeah, pilot, when we're done with this, getting a close-up view of the white, can we go back to the corals and get some more images? Why are you laughing?
It was a beauty. Not yet.
Well, especially if there's a corallium, I'd be really interested to go back.
Yeah. And then we'll...
That's me. Okay, go on in a little bit more. Hold.
That looks like ice, doesn't it?
Nothing's... Okay. All right, full wide. Sure,
I've already told
promising.
Can they get a zoom on full zoom on that white area right there, if possible?
The area that we were just over.
No, if we want to get that
pilot, they're asking for a zoom on the white area, but they're a couple of seconds behind, so
yeah, 7.
It may have been when you panned over.
Okay.
Yeah, it looks a little bit like the what Carolyn and I were calling the marshmallow fluff up on one of the other sites, just a little bit north of here.
Similar ahead, but
yeah,
this is definitely...
It looked a lot icier to me than
yes.
even what we had seen earlier in the day, what we were calling that marshmallow fluff.
Yeah, now one thing is that if we actually hit class
I mean, we're 10 seconds behind, but he's still pretty close to it if he has the chance. I know the vehicle's moving a lot if he's got a chance to study it.
No, he's going back.
Okay, yeah, he's on it.
See, the coral faction pulled me off, but the clathrate faction has regained my attention, and here I go.
I was actually trying to do both. Where are you? And then I got distracted by other voices coming in. Mute.
Looks like pretty good distance, Dave.
Hang on. Let me get steadied up here. Okay, go on in.
Zooming in. Can I zoom in, co-pilot?
Yes, you may.
Okay, I'm going to take the lasers right over the stuff. Back out just to where you can see the lasers, please.
Okay, it's a
hold
partial.
Yep, hold that. It's a real litmus test for ice.
Bernie, do you guys have the capacity to sort of map this whole area out with the dead and live and everything, with what you're seeing here and with the RV tracks?
We might be able to put some of it together. Yes, some of it, but I would plot just all of the images, all the snap photos in, and try to do the tracks around here and try to piece something together. It would take, because I'm not a mapper per se, but I would try to get as much of this down as I could to see what we've got. If we could image it with Sentry, it'd be a piece of cake.
And Bernie, one of the things I'll do is I don't think it's going out, is the HD2 camera on the feed, but they might still be recording that on board the ship, and that might be one of the ways that you could sort of map an image at the same time. So we'll find out what the status of that feed is and whether it'll be available like at the end of the day or tomorrow.
I think we're good.
I agree.
For
Brandon,
posting on the website.
That'd be great.
He's busy in a conversation.
Are we good on the imaging on the marshmallow stuff?
We'll clip it.
Yes, it certainly does look like the fluff that we saw at the other site, doesn't it, Carolyn?
Yeah, and we should try to get in as close as we can on this. I know we're having some issues with the vehicle movement, but we should really try to get close. Unfortunately, as I said in the chat room, I'm not sure we're going to be able to really diagnose this until a vehicle gets down there. It can perturb this a little bit, and then we'll know instantly something about this material. But
okay.
So I'm going to end up under you here if I go for this.
We've got a lot going on right now. Yeah.
Hey, Rowan,
I think,
Carol,
we're moving back to the corals right now.
Okay.
Sorry, a lot of things were happening all at once. I apologize.
And we saw it. Both Roland and I saw it moving. So suggested that was more of a fluffy material than.
Oh, I lost it.
I agree. I saw that as well.
And we did go in super tight, and we're able to see those.
And we did get pretty close to a max zoom, if not a full max zoom.
Oh, I went the wrong way.
On that.
yeah, we're just seeing that now, so we were obviously we're behind. So
Yeah.
that's great.
You guys are really behind today, it seems like.
That's quite a long lag, yeah.
All right, we'll try and factor that into our conversations
really
here.
Yeah, one of the tricks we found is if you do a reboot, sometimes you get closer to our live time.
Sometimes if you reboot your browser and start, quit it and start over, that will help you get back closer to our live time, Bernie, if you want.
Yeah, we'll go ahead and run that. We'll do that. Thanks.
Okay. Thank you.
Yeah, right now I just did that. We're just seeing you come up over that large coral.
So. Okay.
Good. Pretty close.
So now you're pretty much real time with us.
Yeah.
Yeah. So.
No, I'm just gonna swim through here pretty quick, and because I'm sure it's been clipped.
I think this one has. There were some more. I think north along this ridge.
Zooms on associates.
And
there's
the
a white thing
corallium
on the back that I didn't see before.
Would you like to see it now?
Yeah, let me get a little closer here.
So we're imaging Paragorgia right now, a type of bubblegum coral. We're at a depth of 1421. Our local time is 1430.
Okay, let's come in from here, Roland. Hold that. It's just really white.
It's an enemy.
You got more zoom?
Yeah, I'm just getting my focus.
Okay.
Blacks.
It's an anemone. Yep.
Okay.
And some bear skeleton at the base.
Walk it up on this.
The depth is 1421 meters.
There's another one right.
2 anemones on this Paragorgia, and at least 3 brittle stars.
And farther back is another brittle star, I think. Yeah, the disc is obscured.
Yeah, the discs are on the other side.
Okay, let's just work around this way. Okay, come out halfway, please, Roland.
Cup coral there too.
A little bit more. That's good, hold that.
Losing light.
Yeah, I'm gonna shoot around the other side of it before I have to go.
So the ship has stopped moving, but we're swaying. Still got some sway left. Those bubbles coming up?
Does appear to be bubbles in the Sirius view.
Yeah, we got bubbles right in front of
And we're coming
bubbles
around.
Or just crud that I've stirred up?
Yeah, it's shiny.
Okay. Well, just mark the position and take me back there in a second here.
From Sirius.
Gaddiropsaurus. Numerous desmophyllin cup corals. A red crab.
Kind of at the end of the tether here. I'm getting bobbled all around.
I can come down a little bit. Actually, I can come down a decent
I think I just saw bubbles come from you, Dave, bottom right.
There's something blinking in my lights there.
Oh yeah, there's a
you can
good
back
stream.
up a little bit.
Back up a little bit.
Okay, it's gonna be zoom
They're
out.
right underneath you.
Oh, come on, I was just here. But
I know.
But there they are. Okay.
Before we go to the bubbles, is it possible before you leave to get images of these brittle stars? Sorry, we're here.
You know, you just can't wave the big pink coral in front of their face and let it go.
No, you gotta get. We have to get close up to the disc
because you brought us to the bubbles.
Somewhere in this
So we
area.
found the bubbles by coming around this coral and trying to get zooms of the brittle stars.
You hadn't seen the bubbles before?
No.
This is the first time we've
no,
seen
we haven't seen the bubbles yet.
Well, stay on the coral.
Well, that's pretty cool. You just get the corals in the bubble. Oh, look at that. Yep, there they are. Awesome, guys.
You see them? I still don't see them.
They're a little bit on the right.
Serious.
Yeah.
Yep.
Can I zoom in serials to the bubbles?
It's all right with me.
So, a question for you, Carolyn. I would suspect that it may not be enough of a venting of the methane to be changing. Do you think it would be changing the DIC of the water, like right around, within a few meters of the sea floor? And what I'm thinking is, would you expect to see some of that signature in the carbon isotopes of like the corals that we're seeing growing so close to the
next slide.
The mussel beds?
Okay, we can zoom in a little bit here, Roland.
You know, I'm the wrong person. You would have to ask my geochemist about the DIC and what it's doing. Because, I mean, who knows? That's a great question. I honestly, in the seeps I've seen, and others would know better than I've not seen the corals growing this close to the actual venting, obviously. We normally see these other organisms that we're seeing in the background here more closely associated. Maybe Bernie has something to say about that, but I don't.
Okay.
We have, in the Gulf of Mexico, one species in particular, Calagorgia americana, is actually higher, more abundant at seep compared to non-seep areas.
Hold that.
In the Atlantic, this is the first time I've seen corals this close to the seeps. Now, that's only a handful of seeps that I've seen, so that's not necessarily representative.
Center disc.
Oh.
Well, given that there were only a few seeps until about November of last year, that's why you've only seen a few. So.
My tether situation is difficult here, but if I bottom, I might be able to look up.
Do you have any ideas, Bernie?
So take me full wide if you would, please.
Yeah, I was just looking, when we were in Barbados, looking at some of the steep areas down there last summer, we did not see any of the corals near those areas at all. We would, because we were doing some exploration out and around, we would find them, but they're quite a distance away from any of the seeps that we found.
Yeah. Well, the reason why I was asking is because I remember a paper, and I don't remember the results of the paper, but a paper by Jody Smith looking at trace metal signatures of deep sea corals that were collected near hydrothermal vents. And so I was just wondering if there might be a record of the the DIC pool and the changes in carbon that might be recorded in these deep sea coral skeletons, given their proximity to the seep.
Yeah, I've got nothing on that one.
All right,
Josh, it would help.
Food for thought.
Okay, we can come in tight on the coral.
So we're going to drop a target at the bubbles, and could we get a lat long when we get back to there? Please.
Absolutely.
There's another small Paragorgia colony below as well.
Okay, Josh, if you could help me with tilt.
What's the flat?
I'm starting to stir up a bunch of crud here.
The lasers are on? What's that?
Yeah, I don't know.
What would you like, tilt up?
Up.
Looks like a big mussel shell, doesn't it?
Looking at the brittle stars. Okay.
It's awfully big.
We put in the dust filter.
Yeah, some
you like that,
trash possibly.
Yeah, it could be trash.
Okay, come in some more.
Nope.
Oh, that's right.
Yeah, you have it now.
There we go. Getting the brittle stars in zoom.
You have more tether now.
Numerous cup corals at the base.
The same way. I gotta come up a little bit though.
There's one. Just barely see that other guy there. Is that max zoom, Rolly?
Nope.
Oh.
Hole. Now it is.
Can't quite make out that one. All right, I'm coming left. There's that one there. Come out a little bit. Let me find the next one.
Hang on, let me get my focus here.
Okay. All right, hold that. Oh, that was it. There's only 3. Are we happy, science?
I'm happy. Is Tim, are you happy with this?
Let's get a full frame.
Okay.
Okay, yep, we can come out. Do you see right below that the dark, shiny object?
Yep.
Can you just get a snap zoom on that?
Zoom.
Rock.
Is that a turtle shell?
It's the shape of a turtle shell.
It's rock.
Can you tilt me a little bit?
But I think it's a rock.
Oh, you think it's a rock?
Yeah.
Just has the shape.
Yeah.
Oh, yeah, it is.
Little guy
Yep.
behind
Okay,
it.
thank
Rock.
you.
Full wide, please.
Little eyes poking from the back.
Yeah. Okay, now about these bubbles.
Okay, well, let's go look at the bubbles.
I would not think that they would be behind me.
They were right underneath you, I think.
Right underneath me.
Yeah.
Yep.
We don't see them anymore. Let me come down a little bit.
Well, luckily we have a nice current to push all this spooge out of the way.
Yep, there they are.
There they are.
Yep.
In your Sirius view,
Look at Sirius.
bottom right of your camera.
Oh, yeah. What are they doing out here?
You about to lose it if you go any further backwards.
Yeah, I know.
Bubbles.
There was a cry from the other room as Mimi celebrated, I think it was.
Probably that sandy, that rocky rubble directly below your lasers.
Sure that fish isn't outgassing?
Well, nearly bare seafloor, I note.
Now I lost him.
I think it's that rubble bottom right of the lasers.
Yeah, headed there.
Noticing serious view. Is that your tracks or is that different rocks?
No, that's my divots. I was landed there.
All right. I am striving to bring out bubbles.
Yes, I do. Copy that.
Zoom in.
Workshop, this is Copilot.
Zooming to rubble.
Jeff, do you mind sitting in nav for a little bit at 3 o'clock?
Black dots.
Yeah, maybe like 5 minutes before 3, either yourself or someone else come relieve Brian for a little bit.
I'm looking.
Can I zoom into that?
Yep, well, not until 5 before 3, but okay. Thank you. Jeff will be right up. You're welcome.
Camera up.
Yeah.
Yeah, I don't see the bubbles anymore here. Strange.
It's the siren. Just lured us here.
We're trying to find a close-up of them now?
Trying to find them, period.
Why are you laughing? I'm sorry.
They just keep disappearing.
We're trying to find them, period.
Oh, okay.
They were definitely here.
Okay, just come full wide and let me put myself in the same position as before. Top of my camera, don't move. Oh.
But you can see them in the water column. Tracing it back to the seafloor might be a bit more challenging. I think you got to swing back left here.
Halfway up the screen.
I think Roland has his eye on them.
Yes, something's happening.
They're not coming out too fast. It's the problem. They're right back there. I see where they're coming out, but I don't know how to tell you to get there.
Yep, I got them in my view too now.
Yeah, there you go.
Yep, Roland caught him.
It's the bubble master.
You got him. They almost.
Okay, coming left.
There they are. I see them.
Oh, way out there. Yeah, hang on.
All right, we have bubbles, methane emanating from the seafloor here.
Oh, this is pretty cool.
Yeah, this is kind of interesting in this type of sediment, isn't it?
Yep.
Yeah, but like I said, we saw before further north up this ridge
so
that they were where we found the
just
bubbles. They were coming out in the middle of a mussel debris field. I mean, it seems like, you know, I wonder if the, you know, the mussels are probably living some places where the methane's more diffuse. I don't know, and then the bubbles can come out the bare seafloor. I don't know.
Okay, go.
Or there's different points of exit from the seafloor.
Yeah, yeah, right. Yeah, so when we've imaged, you know, the few places where we've imaged these conduits, I think I said a few weeks ago, it's like a tree basically under the seafloor, and you've got this dendritic network feeding these different places, and they can come off and on for various reasons. This is very cool.
Is that a bubble just in the foreground, just a tiny bit that's just sort of stuck there?
Lasers.
Yeah, we'll want to get lasers on that too.
Okay, well that'll require some fancy flying. Come on.
One thing I can add. This is Tim back on. Last year in the Gulf of Mexico, we've got really good footage from the little Hercules with the Okeanos.
Wide.
Really nice, strong streams of methane coming out from underneath mussel clumps. It's interesting to see the difference.
I just drive the bus.
I mean, the most robust part of the field when it comes to biomass and mussels is where the bubbles were.
Huh. Yeah, so an interesting question is, you know, whether like, is this an area that's going to get colonized, or, you know, how long does this particular seep stay alive? Does it stay alive long enough to be colonized? I don't know. This is good footage, guys. This is really nice.
Keep the lasers.
There we go. We have our lasers in view.
I'm only looking at one. Are we close to the waypoint? Because I don't have a screen that I can look at that right now.
No, we're about 15 meters north.
If you look at stream 3,
43
you should see
north
the
of
map
the waypoint
and the ROV camera.
Sorry. Yeah, unfortunately, I can only look at one at a time today.
Josh,
Brandon, when you get a chance, can you give us a lot? Oh, you already gave it to him. Sorry, I didn't catch it. Sorry. Got it.
And Carolyn, we have to do an exchange again. We're having a 3 p.m.
Yes. Okay.
Carolyn, look at your stream 3. It is a quad view, includes the ROV map and sonar.
High resolution.
And pilot screen. If you can view that one, it'll give you the most information.
So
thank you. I did check the screen. It's all fuzzy for me, but it's okay. I get where we are. Thank you very much.
I mean, the
Andrea, please do whatever you need to do. I know you got to set up. So cool. I think we have some pretty good video here if you've got something you need to do next.
Anybody else still up on the conference line?
This is Brendan, the OKX
yeah, I am now.
in the forward lounge.
And is the conference line still live?
Yeah, we're doing another live one. We're going to do one every hour on the hour until 6 o'clock tonight. So we're letting them, I'm letting the pilots do imaging, but Carolyn, if you see something that you want done, let me know and we'll convey it, or we'll see if they can fit that in while they're doing the other stuff.
No, I'm fine for now. I just didn't know if I was having technical issues again because it's been so quiet. That's all. Thanks.
I was wondering the same thing. Okay, Shoreside, we're free of doing the PR stuff, so we have a little bit of time before we have to do the next one. So, any suggestions on what to look at, or any more imaging that you want to do?
All right, science, I'm back. This is Andrea.
Let's go see if we can find the corallium.
Science, this is video. Systems have been returned to science mode.
Alright, I see your light pool. I'm at coming to 025. Come down.
Okay, our depth is 1421. Local time is 1511.
Yeah, I think you're still behind me.
Yep.
Yep.
This is where I need to go.
I'm going to come
We're
up a
still
little
working
bit.
the same muscle bed here.
So we've beat this area to death. We found some bubbles where we least expect them. Maybe it's because all the bubbles that come up through this bed get absorbed.
Or eventually blocked off by too many mussels.
Yeah, it's just all eaten up. So
Did
anything that comes up,
did all of that
over there
awesome.
might be more likely to make it to the surface.
Good. Moving over this dense field of several young recruits.
Starting to see your light pool.
Yeah.
I'm at 90.
We're going to do a video change out for a short while.
Okay. Come up a little bit as you come under me. That was some nice flying, Dave. Pretty shots.
Yeah, the current, when you've got a steady current like that, you can work with it, have fun. Makes it look easy. I think that's a big fat octopus. Two of them.
We had so much light in the background that it was great. All right, so what I found with the sonar is if I turn the contrast, the gain all the way up.
Okay.
You can see those.
Where are we, everyone?
Bubbles very clearly.
Okay.
But it's all about the gain.
Have a octopus in view.
Do you remember where the corallium was?
What's with that octopus? Jared.
Two octopus.
Brother Jared, zoom us in, please. I don't think octopi have eyelids. Ooh. Is that injury?
So we've worked this area for quite some time today. I'm curious if we should start moving up north.
No, not yet.
Are we going to plan on staying here?
If we're going to move, we should move. It'd be worth doing another look at that corallium if we can find where it was at.
Yeah.
Have you guys ever seen an octopus like this?
No.
Nope.
Waiting for, we're not in the chat room right now. I'm hoping Mike Vecchione.
Yeah. He's,
But can you keep panning right?
He's the victim of a terrible mishap.
Or zoom into his...
A little bit more zoom, please, Jared. I'm not hearing you, Andy. They've got the buttons all fouled up. Ouch.
Yeah, I don't think he's looking terribly happy.
I think he might be dead.
Yeah.
Let's look for some respiration.
This is Tim. You know, it's interesting when we see chemosynthetic mussel beds at hydrothermal vents.
Not dead.
I'm thinking those that I know around 2,000, 2,500 meters, something like that. You often see that the octopus live beneath the mussels, and you often see them crawl out from underneath hummocks of mussels. Here you see them, at least two anyway, exposed. And when I look at the mantle on this one right here, looks like it's been, I don't know if those scars on the mantle are actually from shells of mussels that they dig in and go after them, and then drill into the valves and break them apart. I don't know. I don't think I've ever really seen a mantle like that. What looks to be like scars
right over here.
Like scar tissue.
Yeah.
And could we get a zoom on the other one as well?
I'm still not hearing you, Andy. I know you're talking at me.
Oh, I'm sorry.
There we go.
Hello, hello.
There's a button for everything. What would you like, dear?
Yeah, or as Les Watling says, it could just be really old.
I didn't hear that last.
She wants a
the zoom on the other octopus at the, on the right.
Yeah, that's where we're going. Okay, let's come in a little bit, Jared. See what we shall see. Okay, hold that right there. A little bit more. Hold. Hey, wait a minute. This one's... Do octopus have eyelids?
Mike Vecchione is noting that they become sessile while they guard eggs.
Okay, but do they have eyelids? I mean, can they close their eyes? Because I see bumps where eyes usually are, but I've never seen them covered before.
And Mike is seeing eggs.
Yep.
I guess they can close their eyes. Have we got enough, Andy? Or what would you like?
I think so. Let me just double check. And he can see an egg attached to the roof between the arms.
Watch your left side there. I don't know how close you are.
Yeah,
oh.
yeah.
Squat lobsters underneath the carbonate.
Scratch the paint.
Okay, that's good. Thank you.
Oh, it's come wide, Jared.
Okay. So, do you all know where that line of corals were?
Yeah, it's all good.
It's behind me.
There was a big white one that we would like to take a closer image of.
White one?
Yeah. I don't know if we can kind of pan around.
Well, maybe.
I can come around to the port.
If I
What
just...
was it? Oh, yeah.
I shoot out to the east.
Oh, you're going to the east. All right. I'm coming.
And then turn south. And it's just down here a ways.
Yeah, headed back north, yes.
South.
Oh, really?
Yeah. I moved north away from...
I think it might have been up there.
I have no idea.
I think it might have been to the north, so we'll see.
Well, wait a minute, was it with this big pink thing, or was it
It was
somewhere else?
it was a line of corals, and we marked it up there. I think it says North Reach. Yeah, coral in view.
Yeah.
There's more up that way.
Okay.
So let's head up to the north.
When you said go back to, my time scale was different than your
I am
time
sorry,
scale.
it's
You were talking about the previous watch.
Yeah, I'm sorry,
other
Dave.
folk.
A little crazy to try to keep up. Apologize.
Floggings will continue.
I will follow you as you come by.
Okay. We were here.
Looks north and a little east from you.
We're passing over where we saw the 2 octopus.
So...
Yeah, to the right.
There it is. It's a big old thing.
Actually,
yeah,
I think
all of them are on this,
yeah.
this front range and into the current here. I'm going to start from this side because it's down current and I'm less likely to
I don't think
clobber them. Would it be one of these?
It is not one of those, but we can still look at, I don't think we've looked at these yet.
Okay,
so we can go ahead and get images of these corals. That would be great.
Oh, and there's a monster of an octopus underneath.
Yeah, there is. Yep.
An image for associates, obviously.
Yeah, okay. Let me just creep up here.
And I bet you're going to have to go to the other side.
Well, I will eventually, but
yeah.
I just like to.
With all the discs are going to be facing that way.
Well, I'm going to need you to come down, Josh.
Can see the eyes on this octopus.
Coming down.
A few Desmophyllum cup corals. This
I think we just lost video feed 1.
You lost video feed 1. Okay.
Yes, I think several locations have.
Did you get that video?
Yeah, I can confirm that. We've lost it.
And feed number 2, stream 2, is just a big lime green block.
Okay. Right now we're setting up to image a few Paragorgia colonies. Oh, I know.
Let's put your lasers back on. Lasers. Look at the base of that guy.
Both red and white morphs of Paragorgia.
Are the lasers faulting at all today?
Not that I know of.
Nice.